Today we're going to be discussing the Maxam-Gilbert DNA sequencing method. This is a method or technique that is used in labs to figure out a sequence of a particular DNA fragment. And I have here an illustration that I'm going to use to show you how this method works. It's a very basic one because I'm not an amazing artist. I wish I could be, but decided to become more of a science guy. Now, what I'm doing here also is showing a hypothetical DNA fragment that we're going to use throughout this tutorial to explain how this method works. One important thing to mention, and to give some good credit to the two scientists that discovered this or developed this method, this DNA sequencing method, and these two scientists were Walter Gilbert and Allan Maxam. I hope I'm pronouncing their names correctly, but if not, give a shout out. Now, what I need to tell you first is that the difference about this DNA sequencing method is that it uses a few chemicals, and these chemicals are able to cleave DNA at particular bases. And that way you're going to be able to create a few DNA sequences that can then be observed or separated in gel electrophoresis and then observed in autoradiography. But I'm going to show you this in this tutorial. Now, the first thing, an obvious thing to do, is to label. So you need to label this fragment here with a radioactive isotope on the first nucleotide. That way you will be able to visualize later on these fragments that you will be producing with these chemicals. Now, I have here 4 containers. On each container has or contains these chemicals that I was talking about. So the first one will contain a certain chemical that will be able to cleave at every guanine. The second one, a chemical that is able to do 2 bases, guanine and cytosine. Guanine and adenine. And the third one is going to be cytosine, and the fourth and last one, cytosine and thymine. And I'm going to explain in a few words how this works, but I'm not going to do for every segment that is going to be produced. I'm going to write them down here, but I'm not going to write them as I go because it would take a few hours. But for now, to explain how this works, so if we're looking at this chemical here, and say if we throw in all of these fragments here, so these fragments are in these chemicals, so we're mixing the DNA fragments here. Don't forget they're all labeled, but I don't need to do the labeling, but you can see that they're all they're labeled by the radioactive isotope. Now, what I want to show you here is that if we're using or looking at the first chemical, every guanine that it will see, then it will cleave. So the first guanine that you see in this fragment, this hypothetical fragment that you're studying, is here. So this chemical will be able to cleave this particular fragment right here and form a fragment that is ATT, and so on and so forth. So if it finds another guanine right here, we'll cleave here, and also since we have 2 here together, we'll also cleave here. So produce two more fragments, and I already have them written down for you so I can save some time. So as you can see, the first one produced was an ATT right here. And then we have the second one being produced, which is right here. So this is here, an ATT, GC, ACC, ATC. I hope the sequence is correct, but if not, feel free to help me out. And the third one is going to be produced right here on this cleavage site right here. This chemical is able to cleave in guanine, so this cleavage site and it's going to produce an ATTGC ACC-ATCG. So happens the same thing on all of these chemicals, these three last ones. And what I can tell you, for example, every time that you find a chemical, in case of this one, where it's able to cleave every time it sees a guanine and an adenine, then what happens is the same thing that I just explained, but instead it will cleave at both. So it will cleave here at the guanine, as we have here, and produce an ATT. And then it would go on further and find an adenine right here and also cleave here. So you would have an ATTGC and so on and so forth. And if we were to study this particular DNA segment and using these for different chemicals, then we would produce all of these sequences, which we will be able to find or go on and study them in gel electrophoresis and also autoradiography. Again, don't forget that all of these sequences, very important, are labeled with a radioactive isotope. After the first step of the Maxam-Gilbert DNA sequencing method, where we broke down that fragment, that DNA fragment that we want to find out the sequence with chemicals that we know where they cleave at, which sites, which bases they're going to cleave at. Now, what we have here is then, or the second step, which is to run those fragments through a gel electrophoresis. And in this case, we usually use a PAGE, or polyacrylamide gel electrophoresis. Now, why we do this? Because we need to separate those fragments that we created using those chemicals based on their sizes. And that's why gel electrophoresis allows you to do that. Now, if you remember well, we're going to be creating a current here with two electrodes, negative and a positive one. And also, if you remember well, DNA fragments are negatively charged due to the phosphate groups found in the backbone. So what also you need to know is that the smaller fragments are going to be traveling further down, while the largest ones will be left behind. So that's how you separate based on size. Now, what I also have here is the sequence that, or the fragment that we are using to explain this method. But again, imagine that at this stage you don't know the sequence. So the sequence here is unknown. What you do know, though, is the number of bases. So you know that in this case you have 13 bases. But now, after running the gel, you're going to end up with something like this. So all these bases. And these bases here, you have 4, as you can see, 4 areas where you're with different bands. And if you remember well, this is corresponding to each chemical. So the first one, first chemical will cleave at every guanine. The second one at every guanine and adenine. And the third one at every cytosine. And the final one, the fourth and final one, at every cytosine and thymine. Now, another thing that you need to know is that I wrote this here in a way that you read from the bottom up. And why is that? Because say if we have a cleavage here, then this fragment is smaller and traveled further down than this one here, that of course is a bit larger than this one here. Okay, so that's why I wrote from you're reading it from the bottom up. I'm going to clear this up so I can explain now how we're going to find out our sequence using PAGE, or this gel electrophoresis. The bands here, as you can see, this one, if we look at the first chemical that was able to cleave at every guanine, so we know that it cleaved here and produced an ATT segment. If you remember well from the first fragments that I was showing you in the first slide, so this is one of the ones produced in with the chemical that cleaves at the guanines. And then, of course, you have another cleavage here and another one here. So you know that at the fourth, say 1, 2, 3, 4, at the fourth base, you're going to have a guanine. So you know that here, fourth place is a guanine. Already, we're starting to sequence this fragment here. Now, going further down, 4, 5, 6, 7, 8, 9, 10, 11, 12. So we know that there was a cleavage here. And of course, we know that it's going to be a G. So 12th position, and also here at the 13th position, another G. So as you see, we're starting to sequence. Now, it becomes a little bit trickier here when we analyze a G and an A, or a chemical that is able to cleave at every guanine and every adenine. And why? Because if we look at this place here, for example, there was a cleavage here, and you could assume that it could be either a G or an A here, right? Because you know that this chemical could be cleaving at both of these bases. Now, how do you figure out that this is an A? Because if you go back to this chemical here, to this area here, you don't see a bend here. So you know that this hasn't, if this chemical, the chemical that cleaves at G, did not cleave at this area here, at this position here, then it has no G here, no guanine. And for that reason, we will exclude G, and of course know that at this position we have an A, so an adenine. And this is how you do for all of these. So you look at these and you start playing a game where you know where these chemicals cleaved, in which positions, and which bases, or relate to which bases they're able to cleave. So, and for that reason, this is how you're going to be able to then reach to this sequence, as you see here. One important thing that I need to mention, actually add to this image here, is that you're going to be able to do all of this because you're visualizing this due to the fact that you labeled all of these. Remember that we labeled with a radioactive labeling probe, and that way you can then visualize in methods such as autoradiography.